Hi everyone, this is the next video for aerobic respiration. And we just talked about glycolysis. But before we move on to step 2, there's actually this in-between step that for some reason people don't want to associate with glycolysis, but it's technically not part of step 2, which is the Krebs cycle. So I call it the linking step because it links glycolysis to the Krebs cycle. Other people call it the bridge step or some other term for letting you know it's kind of in between glycolysis and the Krebs cycle. So if you're using the foldable, you just wrote on this glycolysis section. So go ahead and lift it up. And at the top of this top portion, go ahead and write linking step and then pyruvate oxidation. And that's going to be on the slide as well. Okay, so if you need to pause the video in a second and write that down, no problem. And it's only going to be 2 or 3 lines. It's one small reaction, but it's an important one. So we're going to do linking step here, and then we'll do the Krebs cycle down here where it's labeled. Okay. All right. Let me turn this camera off. I'm going to share my screen. All right. So we have our linking step, and this is going to connect step 1, which was glycolysis, and step 2, which is Krebs cycle. Okay, and so it's just one reaction. We'll talk about why we call it the linking step here in a second. Let's write this reaction down. So at the end of glycolysis, we had 2 molecules of pyruvate. Okay, but it turns out pyruvate is not what's used to start the Krebs cycle. Okay, so we need to make the starting molecule of the Krebs cycle now, so the Krebs cycle can actually happen. And so that molecule has a crazy name. It's called acetyl-CoA. Okay, we're actually going to make 2 of them. And in converting pyruvate to acetyl-CoA, we're also going to get 2 molecules of that stored energy, which is NADH. Okay, that's the entire linking step. But let's talk about why we need this. And so this step is going to happen in the mitochondrial matrix of eukaryotes. Okay, so we're going to go into the mitochondrial matrix, which is where the Krebs cycle is also going to happen, and that's where the acetyl-CoA is going to go in. Okay, so this linking step is happening so that we can create the acetyl-CoA that is going to be used in the mitochondrial matrix for the Krebs cycle. Okay, and for our prokaryotes, this is all still happening in the cytoplasm because they do not have a mitochondria. Okay, so that's it for the linking step. So let's go ahead and I'm going to erase this, and you are going to move to the portion of your foldable or your notes where you're going to just talk about the Krebs cycle. Okay, here we go. And then let me go ahead and change this real quick. So the Krebs cycle actually has several other names. The only other name that I sometimes say is Citric Acid Cycle. There's a third name, but I will never refer to it as that. So don't feel like you need to remember it. Your exam and quizzes and everything is going to reference step 2 as the Krebs cycle. Okay. So just to remember that. All right, so here we go, step 2. So the Krebs cycle is exactly as the name implies, which is a cycle. And so what's going to happen? Okay, we just made 2 molecules of acetyl-CoA during the linking step, but only one acetyl-CoA can actually enter the Krebs cycle at a time. Okay, so I'm going to abbreviate acetyl-CoA as A little c big A for acetyl-CoA. And I'm going to kind of draw it like this. It's entering into this cycle So here's my Krebs cycle, very fancy, I know. I'm going to go ahead and label it just so that on your paper, you're kind of committing this to memory. Okay, so it's a cycle. So acetyl-CoA is going to come in, and then there's a series of many, many reactions that you don't have to memorize. And then it ends by making one final molecule. Okay, I'm going to abbreviate it here, and I'll spell it out in 1 second, called oxaloacetate. So once the final molecule, oxaloacetate, is made, then another acetyl-CoA can be dropped into the cycle. It reacts with oxaloacetate to kickstart the next round of this cycle. That way it keeps going over and over and over again. Okay, so OAA is oxaloacetate. That's the final molecule made during the Krebs cycle that then reacts with the next incoming acetyl-CoA to kickstart the entire Krebs cycle again. Okay, so again, the starting molecule for the Krebs cycle is acetyl-CoA, and only one can go in at a time. So remember, we made 2 of them right in the linking step. So we're actually going to have to go through this process twice. But let's talk about what gets made during this process. Okay, so one of the things that we are going to make an output or a product is going to be 3 molecules of stored energy in the form of NADH. We're also going to make 1 molecule. Hold on 1 second, y'all. Sorry. We're going to make 1 molecule of ATP. Sorry. Yeah. Excuse me. And 1 molecule of FADH2. That's my brain fart is happening. Okay, so again, 3 molecules of NADH, that's stored energy. Okay, 1 molecule of ATP, usable energy, and 1 molecule of FADH2, which is another form of stored energy. So I'm going to write that down here, stored energy. Okay, and so FADH2 is only made during the Krebs cycle. Okay, we've seen NADH made during glycolysis, the linking step, and the Krebs cycle. Okay, so NADH is made much more frequently than FADH2. Now, though, remember, these products here are from 1 molecule of acetyl-CoA, but we made 2 molecules of acetyl-CoA in the linking step. So we need to do this twice. So when you multiply these numbers by 2 for 2 turns of the Krebs cycle, we're actually going to make 6 molecules of stored energy in the form of NADH, 2 molecules of usable ATP, and 2 molecules of stored energy in the form of FADH2. Okay, and again, how did we make this ATP? Just like we did in glycolysis, substrate level phosphorylation. Again, that just means that somewhere in this crazy Krebs cycle, there's a lot of reactions. Some of those molecules were already phosphorylated, so we just snatched the phosphate from one of those molecules and stuck it onto ADP in order to make ATP. Okay, so that is going to be the end of this video, and we'll have another one on oxidative phosphorylation, which is the 3rd and final step, coming up next.